Hello everyone, Nivedita is here by taking a video on the topic DNA fingerprinting. DNA fingerprinting is a topic from the lesson 6 biology class 12 and this is uh, the topic in which we will study about the procedure and its utility. So before going to the technique, we should know what is satellite DNA and what is its role. So what is satellite DNA? It is the bulk of the genomic DNA and these sequences do not code for proteins. So these are specific sequences and these sequences show high degree of polymorphism. Since the DNA from every tissue of an individual shows the same degree of polymorphism, and this is the thing which formed it a basis of DNA fingerprinting. Okay, that DNA from every tissue of an individual shows the same degree of polymorphism. Since polymorphism is also inherited by children from their parents, and it also helps in paternity testing in case of disputes. So satellite DNA is a part of DNA whose sequence do not code for any protein, okay, and showing the high degree of polymorphism. Now what is DNA fingerprinting? Okay, so first of all, the technique was developed by the Dr. Alec Jeffrey, and when he was doing an attempt to find out markers for inherited diseases. This marker word you will study in the unit biotechnology. Okay, and DNA fingerprinting is also known as DNA profiling. And the most important fact for DNA fingerprinting are the short nucleotide repeats. Short nucleotide repeats. You can say this is the concept. This is the concept or the principle of the DNA fingerprinting. The principle of DNA fingerprinting, it is based on the VNTRS. That is variable number tandem repeats. Okay. This vary in number from person to person and these are inherited. So VNTRS is the basis, or you can say this is the concept for the DNA fingerprinting. Okay, means VNTRS are different from person to person. Means this may be the VNTRs of two person may be of same length and sequence at certain site, but it varies at others. Okay, now what is the procedure to get the DNA fingerprinting? So steps are there, and first step is extraction. Extraction means whatever source we have. Okay, whatever source we have, we need to extract the DNA from that cell, and this is done in a high speed refrigerated centrifuge fuse. Okay, this way it is extracted. Now the DNA is extracted. After this, what to do? The second step is amplification. Amplification means what DNA we have to make out several copies of the same DNA, and this is done by a technique called as PCR, that is polymerase chain reaction. Okay, so many copies are made. Now the third step is fragmentation. Fragmentation means the DNA is cut into fragments, and this is done with the help of restriction enzymes. Means restriction enzymes are the scissors. Now fourth step is means now already the fragments it is cut into the fragments. Then what to do? Then require the separation of DNA fragments. Okay, so to separate out all the fragments, a process gel electrophoresis it is used, and in this process fragments are introduced and passed through a electrophoresis setup, and this setup contains agarose gel. And why agarose gel only? Because the agarose gel is having the sieving effect. That's why the agarose gel is selected. So uh, it is means that setup is equipped with the agarose gel. And what happens? The fragments in this gel, it can be visualized by staining them with a suitable dye. Okay, means DNA, they are colorless, so we need to color them. Okay, so this is the separation of DNA fragments. Means the name of the procedure is, or the technique is, gel electrophoresis. Now fifth step is southern blotting. Now what is done? The separated DNA sequences are, these all are transferred onto a nitrocellulose or nylon membrane. Okay, from the agarose gel they are required to shift. So they are shifted to nitrocellulose membrane. Now the next step is hybridization. Now what is done? Now all the DNA they are transferred to the nylon membrane or nitrocellulose membrane. Now the nylon membrane is immersed in a bath and radioactive probes are added. Okay, now question arises, what are these probes? So probes are the DNA segments of known sequence, means small part of a small segment of the DNA. So these probes target a specific nucleotide that is complementary to them. Okay, so this is done in the hybridization. Now next step is autoradiography. The nylon membrane is pressed onto an X-ray film and the dark bands develop at the probe sites, means probes get attached where their complementary sequences are there, and these resemble the barcodes. Okay, so these are the 7 steps of DNA fingerprinting. Now, what is the utility of this DNA fingerprinting or technique? So first, it is used to identify criminals in the forensic labs. Second, it is used to determine the real parents. Now question arises, what are the real parents? Real parents means we are talking of the true biological father and mother if there is any case of dispute. Okay. And it is also used to verify whether an immigrant is really a close relative of the mentioned resident. So for that purpose also this is used, and to identify racial groups to rewrite the biological evolution. So these are the purposes for which the DNA fingerprinting is used.